Okay. Good afternoon. For one of the high spots of the workshop, it's my pleasure to introduce Professor Pedro Valdés-Sosa. Pedro Valdés-Sosa is the founder and scientific director of the Cuban Neuroscience Center. He's founder and director of the Cuban Human Brain Mapping Project. He's a Thousand Talents professor at the University of Electronic Science and Technology of China in Chengdu. And he's the director of the joint China-Cuba Frontiers Research Lab in Translational Neurotechnology. I can almost hear Pedro's voice describing that title. For those of you who don't know Pedro, I'll give you a little anecdote from 25 years ago when I first met Pedro. It was at that time in my lab we were generating statistical probability maps of the distribution of gray matter and white matter in the brain, which at the time was relatively novel. Pedro showed up unannounced with one of his postdocs in tow, who is now a world-famous scientist in his own right, Jorge Riera. And they asked me if they could have those probability maps to constrain the inverse solution of their EEG data. And of course, this was not a problem. They could have the data. And I didn't expect to see them again. 18 months later, Pedro and Riera showed up in my lab again to show me what they've done. And to this day, it's probably the most shocking experience I've ever had in science, because they had collected data from all across the island of Cuba, from the entire population, ranging from age range from toddlers to 90-year-olds. They had all of this EEG data. They'd taken our probability maps and constrained the inverse solution such that they had a map of normal EEG across the entire brain, across the entire age range, across the entire national population. And the maps had spatial resolution similar to PET, on the order of a centimeter, but still retained temporal resolutions of milliseconds. And it's that time I realized I wasn't in Kansas anymore. And for that, it was the origin of what has now become a 25-year relationship between our lab and the Cuban Neuroscience Center. We've had a succession of brilliant young Cuban scientists pass through the lab over the years, and we presently have Yasser Aturi Medina is the latest version of that collaboration. And we're just now hiring Jorge Bosch Bayar from another one of the Cuban diaspora, who's coming to Montreal to work on EEG inverse solutions and analytics. As of today, he arrived in Montreal for this workshop. So I'm very pleased to give you a little bit of background of the Cuban contribution to EEG over the years in the person of Pedro Valdés-Sosa.
So what Alan didn't tell you was that I arrived 3 minutes late to that first interview. And I said, oh, I'm sorry. You know, I know that English people get angry about it. That's where I learned about some differences between Wales and England. Anyway, it has been fun. And what I'm going to do today is that this is not going to be a very formal talk. I'm going to give you some ideas on where we're developing methods. Regarding consciousness, I've worked with people that are interested in consciousness. And what I have done is provide tools for them. And I hope that from this workshop, this can continue. Okay, and I'm always very happy. I spend half my time in China, half my other time in Cuba, and all the time thinking about methods, but also thinking about how to engage these methods to provide population health and how to provide facilities for developing countries to use these. So just briefly allow me to give you some background. This is the current premises of the Cuban Neuroscience Center. It has an MRI facility, a monkey colony. This is the new building we inaugurated in 2014. I'm giving you some information because perhaps it's not that well known. This is an institution that does basic applied research. It has a clinic. It has production facilities. For example, we produce the individualized hearing aids for the whole country using 3D printing. So it's a Cuban style solution for the problem of when you have very limited resources. Then you have to put things together because you don't have, let's say, the leeway to have a long chain to introduce basic research and then pass to do population health. In fact, our centers are measured. This is part of a large complex, not only by the papers they produce, but what health indicators they have contributed to change. Since 2011, and now since 2014, I also have a lab at the University of Electronic Science and Technology, which, in spite of its name, has a medical faculty and has the provincial hospital for the province of Sichuan. Okay, so talking about health, I just want to show you this because this is a global measurement of the, let's say, scientific output of Cuba. Here you have the log of the GDP of 3 countries, and here you have the number of children that die before age 5. This circles show the size of the population. Here you see China, and this type of map, which is generated by Gapminder, is a program that came out of the Karolinska Institute. You can use it and provide charts for yourself. You can see here the history of China. This is the Great Leap Forward. And then this is the US, and here is Cuba. This is the collapse of the Soviet Union, and here you see that we currently have better indicators in this and other parts of health. And I think it's due to 2 things. It is 1, our emphasis on primary health care, and 2, engaging the research establishment to provide tools for this. Now, the history of this is based on an idea that has been evolving on how to provide this, and this is just, for example, one of the diagrams we're now discussing with the Ministry of Health of doing what we call hierarchical active screening, which means that in people at risk, not people who are sick, we try to use at each level ever more sophisticated and sensitive indicators to do screening of any given condition. This is the only cost-effective way to do things in countries that are strapped economically like Cuba. Now, where does this come from? This whole idea of actually doing, for example, hearing loss screening in the country since the 80s and so forth. It comes from the history that due to certain economic restrictions where we couldn't buy computers, we in Cuba developed, here you see the first microcomputer the same year as Japan. And here you see Talia Harmony, the founder of our lab. We then developed methods for quantitative EEG starting precisely around the 70s with Erwin Roy John. This is a guy that I don't know if you know much about him, but he was one of the first people to use radioligands to study brain function, way before PET. He was the guy who discovered endogenous oscillatory potentials in cats and was one of the co-authors of the P300 paper with Sutton. He was also very much interested in consciousness. And in fact, some of the things that I have done of providing methods for consciousness studies have been through his interests. We later went on, and what I want to show you here is the first Cuban, this is 1982, this is the first Cuban EEG system completely constructed, which we then spread out over the country. These are the amplifiers. It uses a microprocessor. This is the team of engineers, physicists, medical people, and many people here. For example, this is Roberto Pascual. This is the guy who later developed Loretta and several variants of Loretta. So the team has been a team that's been very active, and we still work together since the 80s, no? Now, giving you this background, I have to tell you that we published in 1977 the results of the first collaboration in science between the Cuban group and New York University. So here what we were using is multivariate statistics, mainly principal component analysis, and talking about subspaces of features of the EEG that described our normality and described normal people. And there we were already thinking about the EEG being a culture fair, inexpensive technique to look at brain function and, for example, to do prediction of drug response. The article is interesting in that we have many of the same hopes now that we can address with better technology. In fact, I want to show you here this diagram, which I adapted from Alan. This idea that he presented, it's at the basis of Hubble, I would say it's a modern version where now the subspaces are nonlinear manifolds because we now have supervised learning. And now, to study the brain, we have much better techniques. And we have included now genetics. So this is an update that is really a dream. I remember discussing in the 70s with Roy John, wow, if we could get into the same framework, all the imaging and other stuff, this would be great. And frankly, I think the group of people that are represented here, Alan and all the guys, they've made that dream come true. So in Cuba, we started producing these EEG systems, created a network. This is the density of systems over the country. And we started evaluating, using the area under the ROC curve and other quantitative techniques, the value in detecting brain states of pathology, using just the raw EEG. Let me give you an example. This is quite interesting because we organized a congress in Cuba in 1989 where we were able to take the foremost specialists in EEG, for example, Lopez da Silva, who's one of the authors of the famous treatise on EEG, and we asked them to blindly evaluate the EEG and EEG maps. And what we see here is the area under the ROC curve when we actually broke down what was the actual diagnosis, and here we have the standard deviation for different pathologies. It is interesting that with people with chronic alcoholism, all the specialists, this is the average over 7 of the best specialists, they were actually doing worse than chance. They were using the wrong criteria. And this is something that I think that shows, on the one hand, that for some brain diseases, we can detect certain brain states very accurately using quantitative methods. For others, we can't. And this sort of information should guide the improvements in what we're doing. However, we very soon became concerned with the fact that what we're looking at when we record on an electrode is the mixture of different sources. And this is the simplest possible case. We have one electrode, and this guy has only 2 neural masses. Fit in the favorite guy you want to insult. Okay, this is the, okay. So here, if we have this value measured, and that's what I did here, and we have the lead field giving us the coefficients that go here, we can have any one of these combinations of values of currents here and here that will give us the same voltage. This is a very simple visual illustration of what the indeterminacy or idempoteness of the EEG inverse problem is about. In fact, the minimum norm solution is just the shortest distance of this line to the origin. Loretta is the one that has the minimum distance between these two. Here you go. And if you're using what's known as sparseness, or mathematically the L1 norm, you're picking, in this case, because you have only 2 measurements, either this source or this one, which is equivalent to picking 1 or 2 dipolar sources. So any type of inverse solution doesn't have a unique solution. You have to put in prior information in the form of prior distributions or penalties. And basically, the problem is this. Here you go. Let's imagine that we have here a set of generators, a continuous set of generators, G. We have one measurement. We have here the voltage, in this case is a scalar, it can be a vector. The lead field is the linear operator that tells us how the voltages are related to the currents, and then we have sensor noise. Well, to be able to solve this problem, we could minimize this functional, the difference between the observed voltage and any possible current distribution, but this doesn't have a unique solution. So we have to tag on a penalty or prior distribution, and that will give us a unique solution. And depending on how we pick this penalty, that is the particular solution we have. The first one was the minimum norm, developed by Matti Hamalainen. The second one was Loretta, which was developed by Roberto Pascual. We worked 2 years after that. We continued working, and we required that the Laplacian, the derivative of this, its norm, be minimum, but also we required that it be minimum in the L1 norm. Now, what does that translate in? All of these mathematical things translate into common sense stuff. So, for example, requiring that the roughness or the derivative of the current source distribution be minimum in the 1 norm means that you have little bumps and you have zero everywhere. You think there's only patchy activations. Every single one of the methods that are described correspond to a common sense idea. Some of them are actually biological plausible, some are not. The big problem is that mathematics is a complicated and formalized way of saying simple things. And that is a barrier, sometimes, to communication. So, for example, here I give you a table. If you impose penalties that are in the L1 norm, this means is that there's a certain number of generators only. Or in the L1 norm, which is the sum of the absolute values is minimum. Or you have the L2 norm, which is the common minimum norm. You can require it to be smooth or non-smooth, and you get a family of different methods. I call it a toolbox of different penalizers or prior distributions. What I'm going to tell you about is how we've started using some more complicated ones mathematically, but are actually very simple. And that's part of the history I'm going to tell you. So basically, we started developing something that we called Quantitative Eeg, which we then turned into a source imaging method, which is Quantitative Eeg Tomography. From information of the EEG recorded at 128, 64 channels, you can obtain descriptors, for example, of the spectrum, and these can be mapped, and these are the topographic maps, but then you can use one of the inverse solutions to constrained by information, which can allow you to try to find what are the sources of this oscillatory activity observed on the scalp. This, for example, is from 1996, where we found that a very prominent alpha peak was very well localized in a visual area. Now, what was the thing that we said then? Okay, in developing countries and in Cuba, at the time we did our first brain imaging projects in 1990, we didn't have MRIs. So we asked Alan to use one of the templates that was coming out of the ICBM. We used that as a constraint to probabilistically determine where the sources could be, and we produced an EEG source atlas. Here you can see, this is, over here you're going to see it. This is, for example, the average value for the population for very low frequencies up to about 19 hertz. This is the intensity of the spectrum, and this is from age 5 to 95. So if you look there closely, let's see if I can go back to this here. Just a minute. Here you go. You can see how here this alpha peak increases with age and then starts decreasing. It has a trajectory. And we have that for every single box. Now, what did we do? And this is a paper. Precisely, Alan was talking about the first author of the paper, which is Bosch, which came this morning, who came this morning to work with us. We decided to produce images that were transformed by taking out the mean that corresponded to any given age at any given voxel and dividing by the standard deviation. This is what we call the Z image. Now, something that was very curious is that we found that most solutions here, you can see, are really near the electrodes. If you reconstruct what is the point spread function of any EEG inverse solution, you're going to find out that it's concentrated at the electrode system. However, if you compensate by dividing by the standard deviation, you plot this statistic, something totally different comes, and this bias towards being larger near the electrode disappears. I want to show you one test we did of the clinical usefulness of this, because much of this literature is full of simulations. In fact, the term the inverse crime has been coined for this type of paper. People have a model, they generate data, and then they solve the inverse problem with that same data. And that's really cheating. So let me show you something of a data set that's quite interesting, which also shows that you can get better machine learning out of source images than on the electrodes. And this has been repeated again and again. So in Colombia, Dr. Francisco Lopera discovered 5 families that had the E200A presenilin 1 mutation. That means that these people who has the mutation, has 100% penetrance to have Alzheimer about 45 years of age. These families have been studied, and about 15 years ago, we proposed to select with Lopera 3 groups, those that had the mutation, mild symptoms, those that didn't have any symptom yet, and those controls from the family that had no symptoms. And this is the EEG system. Of course, now it's very compact, but that's the one we were constructing at that time, the second generation of what I showed you. And we took this to Colombia. Now, the interesting thing about this was that if you look at the scalp, these here, on these electrodes, I'm not showing all, you have a normal alpha activity for the controls. Theta activity, slow wave theta activity, starts increasing for the group that has the mutation but has no symptom, and then when they get symptoms, it becomes like this. We followed these groups up till now. In fact, we're in active collaboration with the Colombians, who, by the way, want to incorporate these data sets into C-Brain, okay? And they're part of the Latin American Brain Mapping Network. Alan showed the article where we declared that. But this is the sort of thing that is now they can do MRI, etc. But the interesting point here is the following. This is the area under the ROC curve with cross-validated, this is out of sample resampling methods, standard deviation, of those subjects versus the norm, how you can discriminate them when they do have symptoms and when they do not have symptoms. Now, when you take the left temporal, an array from the left temporal region after doing the inverse solution, you get practically 100%. We get exact discrimination. And this is also validated. So, in this case, the inverse solution serves as a spatial filter that allows better machine learning. So, if you do things on the scalp, like detecting states of consciousness, I'm taking this example because I think this is one of the first EEG molecular images, right? And it was very consistent and showed the power of using source instead of scalp. Now, this was then applied by Erwin Roy-John in a paper where he studied 176 people that were being anesthetized, and he got the EEG. So they went in, they were awake, they started counting back when they induced it, they lost consciousness, they started regaining consciousness, and Roy-John, we did this analysis with our source localization. These are the statistical maps compared to the normative data. He found an increase in slow wave activity in frontal regions that appeared, that's this excess activity, and then it disappeared again as people regained consciousness. Well, Roy has published a number of papers on the neurophysics or field theory of consciousness that were based on this type of analysis. I'm just showing the connection of how source analysis actually has been used in this field. We then finally got MRIs in Cuba. We have several now, and we started a second wave in the year 2004 of brain mapping. And essentially what we did was to sample from the general population, gather blood samples for genomic analysis. This is one of our more current 128 channel systems that we produce for the country and for other places, and we did the MRI. And we then did this. This is the sample size. Not all have the images, but this is part of this is what we're now currently implementing. Its introduction into Loris and Seabrain. We implemented automatic pipelines, some of which were essentially brought in from here, from the MNI. Others, for example, this pipeline for looking at diffusion-weighted tractography was developed by Yasser Ituria, and we have this normative database with we then find age-appropriate norms for multimodal integration. Now, let me show you here an example of something about conscious processing of information that comes from patient studies and special statistics, and I think it has relevance to the problem of consciousness. This is, once again, a patient from Colombia, from our friend Francisco Lopera. This was a businessman that had an accident that wiped out his 2 fusiform areas. I mean, it was nearly an experiment. The guy had deep prosopagnosia. However, when you did the galvanic skin response and several other things, he was having covert recognition of familiar faces. And in fact, we saw activation in the fMRI and also in the EEG. Now, having this database that I've told you allowed us to test several specific models. And one of the things we did is to do fMRI guided tractography for, this is a normal subject, this is the patient, and we saw that there was still connections between the occipital face area and prefrontal cortex. What was interesting, this is a paper in Neuro image by the handsome twin, which is Mitchell Valdés, where what we did was to create lesions. So we did in silico lesioning of the normal subjects to create a statistical sample with permutations to do the testing of this. And this is one of the tools with patients that can be leveraged when you have multimodal neuroimaging. On the other hand, this is work from somebody that Adrian knows very well. It's Calixto Machado, where he was so curious about this whole thing that he decided to test in a patient in vegetative state whether there was a significant difference in the EEG spectrum when the mother was talking to the patient or other women were talking. So what we did was a permutation test of a large group of women and the mother. And what you see here is the maximum of the permutation test. This is a significance of activation of the control group versus when the mother was speaking, and this is the localization. I mean, this can be improved certainly, but it showed the 2 things. The interest that the methods had generated among our neurologists, but also the interest that the work of Adrian had generated in Cuba, okay? This is continued. No, there's more work of this sort. Now, I'm coming to a more nerdy part. Where are we going? We've been working in later years in the acknowledgement that electrophysiological and imaging data is not simply vectorial. We usually work with univariate linear analysis, multivariate linear analysis, and at most we work with matrices. So here, to continue and show you the ideas, we have introduced a notation. This is in a review in the proceedings of the IEEE, where to be able to show multidimensional arrays and analyze models constructed on their basis, we use a graphical notation in which the number of dimensions of the object we're going to analyze statistically is a symbol that could be a square if it's known, if it's a variable, it's a circle, and the number of lines that go out are the number of dimensions. So up to now, statistics has been mostly concerned with vectors or matrices. But when we do time, frequency, and space, we're looking at a 3-dimensional object. So electrode, frequency, and time, that's 3-dimensional object. These diagrams, by the way, were developed by Penrose. for the purpose of analyzing space-time relativistic relations. But they've been taken over by the machine learning community because they serve to express complex models in quantum chemistry and even in neuroscience. And that's what I want to show you. So this is a Penrose diagram. Going further, and those that are interested can see the paper. We use notations like this. This is a constant vector. We can say that the vector has to be, or the object has to be non-negative. Non-negative factorizations is one of the secrets, tools, one of the strong tools of machine learning now. And we can say that, for example, if we interrupt this like that we have an orthogonal property along here, like, for example, principal components. Okay, so this is the notational part. We can also represent, for example, the multiplication of a three-dimensional array by a matrix like this. We just joined these guys. So the tensor algebra, which is an extension of matrix algebra, is just a graphical combination. Now, the other thing that we introduced is that you can have objects that are tensors, that is to say, multidimensional arrays, but then they generate, with error, observables. And we introduced this notation, which we can also point to one of these objects and say that we're going to put a prior distribution. This has to be smooth, has to be sparse. So adding this probabilistic information was what we contributed to the Penrose diagrams. And in honor of the Russian mathematician who developed so much of graphical modeling, we call these diagrams Markov-Penrose diagrams. Now, let's get back to what we were talking about. We're talking about the inverse solution. Those guys in machine learning can recognize that this is the usual representation of a model for the generation of the EEG, which has this functional to minimize to be able to estimate the currents here. And this would be our new notation, where we say that to produce the voltage, we have a lead field, which is a matrix. We have here current, which is a matrix. We put smoothness, or whatever I was telling you, as a condition here. And this expresses, as a circuit, a model. Why? Because we got fed up. Everybody has a cute name for an inverse solution. They have it also for the fMRI. And you never know what they are. The statisticians do the same thing. You have the elastic net, lasso fusion. You have the rodeo. It is really something where you really don't know what the guy's talking about. We said, okay, let's write down, you know, like in electronics, you write down a circuit, you put it there, that's your model. We even uncovered that several models were very similar, though the paper, if you looked at the paper, they were supposed to be totally different. Now, why are tensors important? Well, one of the things is that you can take a tensor of 3 dimensions. You can't do that for a tensor of 2 dimensions, which is a matrix. And you can decompose it uniquely into the product, the outer product of 3 matrices. Now, if this were a matrix, we would be decomposing it to one matrix and another, and this is nothing but, say, PCA or something, which is not unique. This is the error term. However, this form of principal components, which is let's say, the principal components of 3 or higher way order tensors, is unique with very mild conditions. This is known as the canonical decomposition. So we have a form of factor analysis. It's like an independent component analysis where we don't have to require independence or orthogonality or anything. Now, how do we represent that? This is the model. We're representing the outer product, say, of frequency, time, and space. We're putting here and requiring that the spatial component have certain properties, and that generates an observable tensor, in this case, the frequency spectrum in time, frequency, and space. Okay, it's not important that you get all the details, but this is important. We can then tag in here the lead field. And this is an important concept. You do not have to do independent component analysis on the observed variables. You can have an observation model and do independent component analysis for the unobserved. So, for example, if you're looking at sources, why do independent component analysis on the electrodes and then try to guess where the sources are? Find the independent components in the sources. And you can put conditions. For example, I was saying the toolbox of conditions. If you require that the spatial distribution be orthogonal and non-negative, that's simply saying that you have non-overlapping sources. So if you say that the sources, you're finding components that are in different places, you can mathematically code that. So we applied that to the time frequency spectrum of the EEG. And for example, we were able to decompose the alpha rhythm. This is just one subject, which has actually frequency peaks at different times. Each of these fluctuate in a different way. So this is the time evolution of this frequency peaks. Sometimes one emerges, sometimes the other emerges. And their spatial localization is like the original thing I showed you near the visual cortex, but actually not only one source, but rather three sources that seem to be tuning in and tuning out. In fact, these sources have a very simple coherence, that is the imaginary correlation, the complex correlation between them at the peak of the alpha frequency, which is like this. There are two that are very tightly coupled, another that isn't. However, if you look at the imaginary coherence, there's a mythology, there's an urban legend that we're able to construct statistical measures that are immune to either the hemodynamic response function or to the volume conduction. That is a myth. There's no way that you know that there's something that's confounding your observations. You can come up with a magical number that will, without taking it into consideration, ignore it. Here, the imaginary coherence was once posed as having no zero-phase component. This is what we found for three components here. This is what happens in 60 electrodes. We actually applied here network analysis and found small worldness. We had a lot of fun with this. But it was the small worldness of the volume conductor. And I don't think that is our object of study. Our object of study is the brain, okay? Not what obfuscates the observations. Okay, so moving on, what is also very interesting is concurrent EEG and fMRI. And we've worked on this using two directions. So the model is that we have an ensemble of postsynaptic potentials that produce at the same time a vasoactive feed-forward signal that produces the bold, or a net primary current density at the mesoscopic level, which produces either the EEG or the MEG. If we use detailed modeling, biophysical modeling, we have a model-driven approach to try to do fusion of these two types of information. Otherwise, we can just do it with machine learning techniques and a data-driven approach. We know that the observation equation here is driven by the volume conductor, which is a smearing, a spatial smearing. And here, because of the way we know bold occurs, we have a temporal smearing, maybe some spatial smearing, which is the hemodynamic response function. And these are the two observation equations that have to be taken into consideration. We all know that if we do concurrent analysis, and the MNI has been one of the places been pioneer here, we have to be able to eliminate the mutual interference. I'm not going to go into detail there. But after cleaning it up, we can see that, for example, this is for this is for several seconds, the fluctuation in the peak of the alpha rhythm with the bold response in, say, the visual cortex. And it has a very high negative correlation. And this is something that shows that you can do some type of modeling in this sense. So what did we do? This is an example that we took where we have here, for each of the electrodes of the EEG, a time frequency plot. And here you see the alpha rhythm coming and going, waxing and waning. This happens for each of the electrodes. Here, what we have is the Z transform for each voxel of the resting state fMRI. And the problem is to look for relations between these two. So I'm going to talk about a data-driven approach that's based on the models. This is a data-driven approach where we put into the diagram both the bold response and the EEG, and we put in determine certain priors. This is a more graphical way of looking at it. We're taking the EEG spectrum, which has frequency, time, and space, the derivations. We decompose it into what we call atoms. And these atoms have a signature for time, for frequency, for space. That's what I will show you. But at the same time, we then look for the components in the fMRI that have a spatial and a temporal resolution. And this is indeterminate. This is not. But if we require that the temporal evolution of the EEG spectrum be maximally correlated with the component we're searching for in the fMRI, then this problem becomes well determined. So here, I want to show you the results. This was one work where we found three main components. One was in theta, the other in alpha, the other was higher, was really not very significant. The localization was totally different. The theta one was more frontal. The alpha was where we've been finding alpha activity, and they fluctuated. And here you see the evolution. So this is the spectrum. This would be the frequency signature. This is the spatial signature. And here you see the temporal signature. And what you have here is the segments in which the individual was at rest and segments where he had to do backward subtraction. So this is very clear relation between these components and whether the subject was at rest, the state of the subject, or was doing a task. We furthermore then did a grandeur causality analysis and found that the task modulated the alpha component, which then had a negative influence. This was negative and negative on the theta component. So this is a way of a second problem, because up to now there's one problem, which is, can we detect with the EEG changes in brain states, consciousness or others? There's a second, can we localize it? Third problem is, can we tease out the neural systems and their interactions that, and then there are methods to do this, imperfect, but there are methods to do this. So, do we need the fMRI? No. We think that the fMRI can be used as a calibration tool, and this is very recent work. This is actually going to be presented at the next HBM meeting by one of our PhD students, Joanna Perez. She's under the advice of Valia, who was a long time a head of department at our lab. Now she's in Aston University in the UK. And here, what we've done is have a subject either listen to a tone, listen to music, hear a message, or hear the message, which is then played backwards. The point that we want to make here is that if you look at the fMRI activation, if you look at the concurrent inverse solution with 60 channels and with 19 channels, the patterns that are emerged are very similar. In fact, this is work in progress, but they're going to take to see how well you can classify all the subjects in which state they were. But this points to something we believe. Properly guided, EEG can probably perform well enough in certain situations. And EEG fMRI, that's very costly, concurrent EEG fMRI, can be used to calibrate where. This is an idea that Talma Hendler has said. She has this concept with Hassan that you have to do EEG fingerprinting. You do EEG fMRI, and you do it to get out what features of the time frequency evolution of the EEG can predict certain brain states. Now, the other thing I want to say is that all of this requires experimental verification. And we're engaged now in a project. This is my postdoc, Qingwang Vincent, from China. He was just in Cuba now. This is Pedro Valdés Hernandez, who did his PhD at our center, is now with Jorge Riera. And we've been working with a model that was created by Onotaka Fuji in macaques. He has implanted ECOG for 128 channels, also covered everything else and implanted the 1020 system again. Here's a bit more of detail. So this is the website anybody can get. This is open science. This is the operation, once again. What we've done is that we have constructed detailed finite element models of the volume conductor and for the positions of the ECOG and also of the EEG electrodes. And we're in the process, we have several papers that are simulations of comparing EEG and ECOG to actually test which are the best methods to try to get at the underlying states. The interesting thing about this data is here you see the time evolution over here of this data. Here, this is the monkey is awake under anesthesia. This is the EEG spectrum, and this is the ECOG. So this is time and frequencies. This is the time frequency oscillation. They're not exactly the same. Here, interestingly, is the cross spectrum of the EEG in awake and anesthesia, both for EEG and ECOG. They are different. We're not surprised because volume conduction is at play. However, what is very interesting, that if you do a canonical correlation, and this has a lot to do, this is in the frequency domain, finding the linear combination that gives maximal coherence between electrodes on the scalp and over the cortex. You find that many of the different components are highly significant. This is the Bonferroni level for all these frequencies, which indicates that there's a tight coupling with it. This is not surprising. So it's our task, and that is work in progress, how to transform this information so that we can improve and actually predict the networks that are inside the head at the cortical level from the EEG. And I'm just saying this is also like the previous presentations, work in progress where help would be appreciated. Alan has already mentioned that this is done in the context of a collaboration with the Montreal Neurological Institute and China. The three agencies have funded joint work. In fact, we are now here for a month to push this collaboration, and we are working on putting on Compute Canada data sets that are so large that they can't be hosted in any of our countries. Now, what do we want to do with these? We are putting in place pipelines so that people in Colombia and Argentina and Mexico and Cuba, even parts of China, can interrogate these databases. This is our dream. Get the results we need and then download those results only to answer scientific questions. And the pipelines I've showed you will be put up there. Finally, well, these are the people I've worked with over the years. These are some of the papers. But I want to end by mentioning this. In 1989, Roy John, who was very interested in consciousness, he invited a group of people that included Herman Hacken, the developer of synergetics, which is how to describe complex nonlinear systems, Lopez da Silva, we had Pribram, so it was a multidisciplinary group, and we wanted to discuss, it was a workshop on how to tackle consciousness, okay? So there's a certain recurrence of history. It was very useful. We kept in contact over the years. Later, I asked Roy to change the title and to talk about mechanisms, no? And put it towards, and we got this book out. But I hope that this workshop is only the first step of something like this that created lifelong friendships and enormous collaborations. Thank you. This is what I want to present.